Today we are going to study the protein structure. In last class we have studied about primary and secondary structure. Today we are going to discuss tertiary structure, what it is. It is the three-dimensional conformation, and that is the overall folding pattern of a polypeptide chain. And the primary structure of a polypeptide chain determines its tertiary structure. That means the sequence in the primary structure is the basis for the tertiary structure, three-dimensional conformation. And what happens in a polypeptide chain? There are amino acid residues which are placed far apart, and they come together and make bonds, and then interacting with each other, they form a tertiary structure. And this tertiary structure represents the lowest energy and greatest stability state of a polypeptide chain. A domain. What is a domain? This is a fundamental, functional, and structural unit. And it is because in the tertiary structure, so it is three-dimensional structural unit and having 40 to 400 amino acid residues. There are large proteins which are having clear, defined, independent functional units. Some regions which are having specific functions. And the core of domain is built from the combinations of supersecondary structural elements, that is motifs. The hydrophobic chains are inside in the interior, and hydrophilic groups are on the surface of the molecule. Here you see this is the SARS-CoV-2 structure. There are different proteins. You are seeing spike protein, caspase membrane protein, envelope protein, and single stranded RNA protein. You see in the spike protein there are multiple polypeptides, and you see there are different domains. Two domains are you can see one is receptor binding domain and one is angiotensin converting enzyme to receptor domain. That is how they are functionally independent from the other part of the polypeptide chain. Simple proteins are having only single domain, but large proteins are having multiple domains, such as lysozymes or triose phosphate isomerase or myoglobin having only one domain, but lactate dehydrogenase is composed of two domains, that is N-terminal and C-terminal domains. N-terminal is for NAD binding and the C-terminal is a pyruvate binding domain. And these folding of a peptide chain forming a domain occurs independently of folding in other domains. So each domain has the characteristics of a small, compact globular protein, but structurally independent. There are different forces which stabilizes the tertiary structure. These are disulfide bonds, hydrophobic bonds, hydrogen bonds, ionic bonds, and van der Waals bonds. And all of these bonds stabilizes the tertiary structure. Disulfide bonds, as you know, by the two cysteine residues and the linkage of sulfhydryl group. And this provides the stability of protein molecule and preventing its denaturation in extracellular environment. Example is immunoglobulins. Hydrophobic interactions, non-polar side chains in the interior and polar and charged side chains are on the surface of the molecule. And in non-polar environment, just opposite occurs. Hydrogen bonds, serine and threonine having a hydroxyl group, and they make a bond with oxygen of carboxyl group of some peptide bonds such as glutamate. And formation of hydrogen bonds between polar groups of the surface of proteins and aqueous solvent enhances the solubility of the protein. And number 4 is the ionic interactions. Negatively charged carboxylate group and positively charged amino group make bonds with the different amino acid residues. Van der Waals interactions, these are very short range, weak interactions, develop only when atoms are packed very closely to each other, but they serve to bring the atoms close and squeezing out gaps, and thus contribute enormously towards the stability of the native protein structure. Now come to the protein folding. In different protein structures, we see there are multiple foldings. So proteins are conformationally dynamic molecules and they can fold into their functionally competent conformation in a time frame of milliseconds. That is the speed of their folding. So the interactions between the side chains of amino acids determine how a long polypeptide chain folds into the intricate three-dimensional shape of the functional protein. And these foldings are done by hydrogen bonds, hydrophobic interactions, disulfide bonds. There is one Anfinsen's law which says knowledge of the native conformation is specified in the primary sequence. As we see in the beginning, that the primary structures decides the tertiary structure, and this is the same. The primary sequence is having the knowledge of the native conformation of the tertiary structure. The protein folding takes place in an orderly, guided fashion and result in a correctly folded protein. There are two steps of protein folding, first stage and second stage, and these are also known as first segment and second segment. What happens in the first segment? Newly synthesized polypeptide emerges from ribosome. Short segments fold into secondary structural units. You see here is a human cell, nucleus, DNA, transcription, processing, messenger RNA, translation, and from the ribosome, the synthesis of a polypeptide chain. And from here, the folding starts. Second stage, that is second segment, the hydrophobic regions segregate into the interior and secondary structure rearrangement till the mature conformation of the protein is attained. And this process is orderly, but it is not rigid. There is flexibility. And there are 4 different shapes during folding protein molecule takes. That is number 1 is domain formation, number 2 is molten globule formation, number 3 is native conformation, and number 4 is oligomeric formation. Sequence specific conformation. Sequence decides the specific conformation. This dependence of conformation on sequence is significant because the intimate connection between conformation and function. The conformation decides the function, and according to the function there is conformation of a protein molecule. And these individual protomers tend to fold before they associate with other subunits. There are 2 enzymes which take part during folding process of a protein molecule. These are disulfide isomerase and cis-trans-prolyl isomerase. This protein folding is essential for the biological function of the protein. Now come to the assisted folding. Sometimes some proteins need assistance, and for them folding is a facilitated process, and for this facilitation they need a protein molecule which is known as chaperone proteins and which function with the ATP hydrolysis. and these chaperone interact with partially folded or improperly folded nascent proteins to facilitate correct folding.